rushing off to do things. So this PhysioX is found in a similar way that the other one was found, meaning that you'll go into Mastering A&P, you'll go into My Study Area. From My Study Area, you'll get which is a menu on the left side, you'll see My Study Area. And then you'll get a menu, and near the bottom, it'll say PhysioX. And so I wanted to share my screen, which I hope I find the right one. Launch meeting, that's not what I want. I think it's this one. That's not that one. Ugh, what happened to it? Too many friggin' screens open. Maybe it's behind this one. Damn it. I had it opened for you guys. It's clearly not that one. Oh, here it is. Okay, so why this didn't show. I'm going to stop sharing that and try another share screen. This must be it. Yep. Okay, so once you get into PhysioX from the study area here, you're going to again go into exercises. And then remember, this is how you scroll down and you did the urinary exercises. Now, right under that is acid-base balance. So the one I assigned is hyperventilation. So hopefully after the last part of the lecture, what that means is somebody's coming in a panic attack, say, or a highly emotional state, and they are expelling large amounts of CO2. So if we go into that one, it's not a very long one. I think there's 10 pages to it. Once again, there's some introduction you can read, and it'll eventually lead you to a little machine, kind of like we had in the urinary system. And down here, there's a box where you're going to record data. So like before, you'll walk through this, and it allows you to change their type of breathing, their patterns of breathing. And these are the little lungs here. And then it records the pH, and as you'll see down here, it records their CO2 levels. And what they mean by minimum and maximum is, during the period of time that they're going to be breathing, because they breathe over this period of time. Here it's about a minute. What was it at the lowest level, and then what did it change to at the higher level? So go ahead and do this, and then again, I encourage you to do this by working in a group, although this one, I think you should be able to answer. It's a little easier than the last one. And the first part is this activity one that is on hyperventilation. Therefore, it's clearly a respiratory issue. And then can I go back here? I think I can. And then you're going to do activity 4, where you have a metabolic issue. So this will give you guys a chance to try both of these 2 little activities and answer the questions in the handout. So the handout has been up there for a while. I did notice that somehow I didn't refer to it in the study guide, so I'll make sure that is referred to in the study guide. I only refer to the urinary one. But the handout is in your materials for the Lab Test 3 folder. Now, this isn't due until next Tuesday, but the urinary one is due today. So why don't we come back? Why don't I give you what we have till 12:45? So why don't I give you an hour to do this? And then at 12:15, we'll come back together. We can go over the urinary one that's due today. Make sure you've got some good answers for that. And if we get through that quickly, we could do a little bit more review. And what I also posted, because they weren't in the PowerPoint slides, was a couple practice patient values. So I, gave somebody a pH, a PCO2, told you it was going up or down, HCO3. I made 3 of those in the past, and I posted those just during the break we had. So that would be something we could work on too, either today if there's time, or next Tuesday if you'd like, just to come up with the answers of those problem sets. So let's see let me stop sharing this, and hopefully if there's questions about how to get there, let me know. But what I'd like to do is put you into some breakout rooms. And again, I'm happy to put people who want to be in breakout rooms together, and those who don't care, I can just manually just pick you together. Check that everybody's out of the breakout rooms. I think they are. Yeah. So I thought the first thing we do is go over what's due today, if that's okay. If there's something else you want to do, we can, and we have time, we'll do that as well. So hopefully most of you finished the acid base, the 2 activities in that one. And in the beginning of, probably what we should do is do it at the beginning of lecture next time, because it could be part of our review. We could go over that and the problem sets for that I set out for, I think there's 3 patients or 4 patients. And again, I was just going through this with Noah. What we're assuming is we're just going to look at this. If the respiratory system can compensate, we assume compensation is going to happen. If I want to ask you then, what would the renal system do to this? It would be a separate question. So we can play with that too with those patients if you'd like when we come back. So how do you guys feel? You want to do the Physio-X for the last one, the glomerular filtration one? Is that good? Just give me thumbs up or something out there if that's, okay. And remember, that's what's due today at 5. The acid-based one, you're welcome to turn in, but it's also useful to do it after we've gone over it. So I am screen sharing the version of this that's blank. So hopefully you're seeing that. Now I didn't put the date in here, although I do have an example of one that I did. And what you'll see is that what they're doing here is they're first decreasing. I think it goes down to the first 4 of them. They keep decreasing the afferent arterial. And hopefully what you saw is as they do that, glomerular filtration rate starts plummeting, really. By the time it starts, I'll put some numbers in here, although this is my original, 0.5 to gets to something like 0.35. So we're not talking a big diameter difference here, and yet you see the rate go from something like 59 to 11 in that, or 0. It actually goes to 0 at 35. And also the amount of urine produced. So it's vasoconstricted down to the point where glomerular filtration is so slow that if you looked how many milliliters per minute, it would be basically none. I don't know how realistic that is, but that's what this model is giving us. And then we went ahead and dilated it beyond, made it larger than our starting point. So I think it goes up to 0.6. And it more than doubles the glomerular filtration rate, and it goes up a good amount for the 30% or something goes up from 162 to 196 from the amount of urine produced. So is there any questions on what's going on with changing the afferent arterial and how that's changing glomerular filtration rate? And is this basically what people found? Again, you can give me a thumbs up or yes or whatever in your reactions or in the participants button. And I'm not seeing anything in chat. Did nobody get any of these numbers? Am I all alone doing this? No thumbs up?
Dr. K, do you think you can go over, like, I think we're okay with the values in the tables, but maybe like the questions to understand the concepts?
Okay, well, that's why I'm doing this, because the concept of this is that as the afferent arteriole constricts, glomerular filtration rate drops, and we didn't change the HP capillary, and the urine produced drops, and then if you go wider, it's going to increase that. So, okay, so then it says explain why it decreased. So anyone have an answer for that? Why would it decrease? What's happened to which variable? Let me put some variables down here. HP cap. I'm not going to do the OP cap. Is it cap? Osmotic pressure. My brain just went out. And HP glomerulus. I think it's glow, right? And HP glow. Which of those changed? Anybody just put it in chat? I'm sorry, my dog is getting a little unhappy with me. HP glow, that's right. So this is a reflection of the hydrostatic pressure coming in due to more blood coming in. So if you lower the amount of blood going into that capillary bed, then you're going to lower the pressure of the blood in that capillary bed. These guys, or at least this one, doesn't change at all because that's dependent on largely on the albumin that's in the blood. And then this would probably, if anything, as filtration dropped, be less of a pressure, but this is the main one you're changing. So that's the right answer. So you can write that in whatever words you'd like to explain that. All right, now we're getting to some of the substances. Name two substances that would decrease the afferent. So what could cause these four things to happen? So just somebody type in a substance and we'll talk about when it's released. Okay, we got norepinephrine, we got epinephrine, we got angio2. Okay. And when would these be released? Well, extrinsically, right? These are extrinsic regulators for the most part, and they're going to be released when mean arterial pressure drops in order to lower glomerular filtration rate so that you can retain more water in your body. So there's a bunch of things that decrease the afferent arteriole that are extrinsic. How about if I wanted to pick some intrinsic ones? Which intrinsic ones would decrease the afferent arteriole? Right, ATP, adenosine, I can't type, adenosine from the macula densa, and they'd be released when sodium chloride goes up, right? In the filtrate. And why that increases is because glomerular filtration rate is too fast, so that sodium isn't all being pumped in with its partners, like the bicarbonate we talked today, or the glucose or amino acids, whatever you're trying to reabsorb in the filtrate in the PCT. So since it wasn't picked up, it's left behind, and that means filtration rate is too slow. So there are intrinsic and there's extrinsic. Okay, so name two substances that would increase the diameter of the afferent arteriole and the conditions. So let's again do extrinsic first. What's in some extrinsic signal that would tell the afferent arteriole to increase diameter and therefore glomerular filtration rate? Anybody can just type it in. Yep, that's right, AMP. And this is released when blood pressure is very high and you're trying to cause an increase in glomerular filtration rate, increase in loss of fluid into your urine to bring your mean arterial pressure down. How about intrinsically? What could we put for that? PGE2. And this is released when, the kidney's kind of trying to save itself because so much vasoconstriction has happened so strongly that too little blood is entering into the tissues of the kidney, and so it tries to, and it is released by the macula densa, or other kidney. It's the macula densa as well that releases PGE2, as far as I know. All right, are we good on this part? Any questions? Okay, so let me look at I won't go through the exact numbers, but so the next part is not about the afferent, it's about the efferent. So this is think of this as the drain on the tub of the glomerular capillaries. So if we left the afferent the same, therefore HP glow is the same, but we slowed the drainage of the blood out of that capillary, it would actually raise HP glow indirectly. Instead of doing it by how much blood is coming in, it's how much blood is exiting. So they kept decreasing this, and what happened to glomerular filtration rate is it increased and urine production. So you can almost think of it, fluid backing up in the capillary bed, and therefore blood backing up, therefore pressure going up in that capillary bed. So name a vasoconstrictor that's used when systemic mean arterial pressure rises and it acts on constricting the efferent. So let's see, there's a question in here too. I'll come back to that in a minute. Has anybody got an answer for who would constrict the efferent arteriole and therefore raise, drop mean arterial pressure? It's AMP again. So AMP is kind of so this guy is AMP. And once again, what it's trying to do is by constricting, having a greater filtration rate, lowering the fluid level in your body. And what does that one do to the afferent? Well, we just talked about that up here. It's a vasodilator. So now you're kind of getting it two ways. You're having the faucet flooding blood in by dilating the afferent, and then you're constricting the drain by constricting the efferent, and therefore glomerular filtration rate goes up. So the question was, and this is my mistake, the program had to change these to 90. So it doesn't change. It's not our variable. I should have typed 90 in here, but don't worry about that because it's not our variable. We were keeping all of that the same. Okay, I think I just went with what was on there originally. I'm not sure why they wanted you to change that, except maybe a later program lets you do it. Okay, all right, so we finished that. So we went on to activity 2, and this is the effect of pressure, and that's what I said the next activity is going to let you do, vary that pressure. Okay, so on my data, we had sort of systematically changing this pressure and then watching how it changed this pressure and change the filtration rate. So right here, since diameter of different are not changed, not shown in this table. Oh, I see. It was just notes to myself. They had more things in your table. Okay, and then I don't know how realistic this is, but they had like a valve at the bladder. I think they talk about it. Maybe a tumor could close that. So in the second part, they actually Don't let the fluid drain out, so it would increase which variable if you close this valve? Anybody can type it in. Is it OP glomerulus, HP glomerulus, or HP capsular? If you close the valve. Not getting anything. HP capsular, right. So that's the idea, that you're trying to, close that valve and cause such a back pressure that it's hard to filter because there's so much back pressure on it. So as they increased the beaker pressure, this is proportional to HP glomerulus, which means you ought to get an increase in glomerular filtration rate and urine production, which they do, and they drained it out here. All right, hopefully. So what intrinsic regulator is being ignored using this little mechanical bottle? Any answers to that one? There you go, myogenic. Yeah, so this makes no assumption. It's almost like you could just have a hose which has no response except to be a tube for that afferent arterial. When that's not the case, the smooth muscle is trying to keep sort of a set point amount of stretch, which is part of that intrinsic regulation. So if you suddenly increase pressure in this, like say you suddenly had a big, strong espresso coffee and made your heart beat really hard, your pressure surged just momentarily, it would stretch those smooth muscle and they would try and return without any kind of extrinsic signaling or intrinsic signals, the chemical signals. The smooth muscle itself tries to go back. So it would help prevent surges in pressure from affecting glomerular filtration rate. Okay, name another part that releases intrinsic regulators. And so this is what's the other active part? Macula densa. So this is a little repetitive from what we did before, but it's okay. It's giving you it in another way. And it could release, and you could choose what you'd like. It could release the ATP, adenosine, or it could release PGE2, and then the situations which go with each. All right. Predict how an increase in osmotic pressure would affect glomerular pressure. So if you increase osmotic pressure, let's just increase the osmotic pressure. Somehow we magically wave a wand and OP goes up. What would happen to glomerular filtration rate? Increase, decrease. Yeah, so it would decrease because net filtration pressure would decrease. So if we go back to our formula, HP glow minus is subtract from HP glow the sum of the osmotic pressure and the capsular pressure. So if you increase your osmotic pressure, then don't change anything else. Your glomerular filtration rate should drop because you're unable to push things out as effectively, and urine volume ought to drop. Ultimately, though, that means your blood volume would begin to drop, right? Would begin to increase because you're not losing as much fluid. So ultimately, HP glow tends to go up in response to that. And probably the way I've worded this should be the other way around. It should be OP glow does this, and then how does HP glow affect? Okay, missed the first blank before GFR. Yeah, probably this is the first draft of this. I should have said, how would the increase in value of this affect GFR and urine volume? And then subsequently, how would it affect HP glow? So if you increase OP glow, you're going to decrease GFR and decrease urine volume. That's going to then later cause your blood volume to go up, which means your hydrostatic pressure is going to go up, and ultimately, not intentionally, but ultimately you will start increasing GFR and urine volume production, but your blood pressure is going to be high trying to do that. So I guess this one needs a little bit of editing. Is that clear? Sorry about that.
Yeah, so there shouldn't be anything in there.
Oh, no, there should. It's just, I can't edit this, but this part may be cut out, and maybe I can edit it. Yeah, I can edit. Answer that and then say, then how would that affect HP glow? That's a probably better way of wording this question. So once you decrease your GFR and your urine volume decreases, your blood volume goes up. So ultimately, that's going to affect HP glomerulus by causing it to go up too. Does that make sense?
Yeah, I think so. I think so.
Yeah. See, if you don't lose as much fluid because you're not filtering it out, therefore you're not making as much urine, that fluid has to stay somewhere. It stays in your bloodstream.
Right.
That means your blood volume goes up. If your blood volume goes up, your blood pressure goes up, and HP glow is proportional to your blood pressure. Okay, so effects of hormones. And again, some of this is a bit, well, not really redundant, but it's still the hormones that we talked about earlier. So they're talking about urine potassium levels, and don't worry about any particular numbers on here relative to what we say the blood ones are, and urine volume, and then urine concentration. So what they're trying to do is vary whether hormones are present or not, and see what happens to electrolyte secretion more than anything. That's kind of what I was trying to focus on in this. It also says it doesn't try to see if there's any interaction, because whenever you move more sodium, you move more water, as long as ADH is present. So it doesn't try to do that. I'm sorry, it does try to do that, show you. It's blabbing, I'm here. After, I guess I should have read this carefully again before we started. So it looks at the independent and synergistic effects of ADH. So here you have aldosterone, neither of these present. So that means that whatever potassium didn't go back in paracellularly in the PCT is probably in the urine and stays in the urine. So we have dilute urine here, which shouldn't surprise you at 100 milliosmol. And the gradient is the gradient. It's always a standing gradient unless you start adding urea from inserting facilitated diffusion carriers with ADH. And then it can go up a bit down here. All right, then we add aldosterone. So aldosterone, remember, acts in the principal cells. So what it's going to do is bring sodium in, but it's also going to increase potassium secretion into the filtrate. So you'll see that potassium goes up. And eventually, enough potassium goes up that you start seeing urine volume also drop, because potassium's an electrolyte, and therefore, things left behind, it's a solute, isn't going to make it as easy for water to go back. Okay, then we add ADH, I think, on the third one without aldosterone. So ADH being added, and you see that, again, the potassium goes way up in that case. So what's happening is its number is going up over here, not because of secretion of potassium, but because water is leaving, and therefore potassium concentrations are going up. The gradient is getting steeper in the medulla, and urine volume drops precipitously. And then if you have both of them present at the bottom here, you again see some increase in potassium, not huge, but you see some increase. That's probably the part secreted. And then you see urine volume drop, and again, the concentration is pretty high. So I'm not too worried about the details of this, but I do think you should understand that they do act to help each other in a way, and that electrolyte osmolarity affects, whether it's sodium or not, can affect water movement, which I think this is trying to show and demonstrate using potassium. Okay, but this part is really important. What conditions or hormones would stimulate the release of ADH? And I didn't ask you to order these as far as which is the most important. So just anybody type in something that will cause it. Dehydration, meaning increase in osmolarity of the blood. Good. All right, what's another one? Angiotensin 2. Yep, that directly. And the last one's probably the hardest one because we didn't stress it very much. Yeah, and I would even say very low blood pressure. Just a small drop or a short-term drop in blood pressure doesn't tend to increase ADH, but very low. And now it's not clear to me if that's directly, like the hypothalamus has osmoreceptors. They never say that. Or it's through multiple pathways where you get greater angiotensin 2 release, or whether it's, somehow there's a sympathetic pathway that will eventually stimulate the hypothalamus. So that's not entirely clear to me either. Okay, now this one I do have you put them in order. So number one stimulus for the release of aldosterone, angio 2. And this is by far and away, so I'm going to say greater than. What's the second one, anyone? High potassium. So that's uncommon. I'm just going to say greater than. I have no idea the amount. Uncommon situation, but you could have it. And then ACTH is the last one. So keep them in mind, this order, because the amount of aldosterone changes with each one of these. And remember, for that essay, which one is it? Where's my essays? Over here. For essay 4, these are useful things to remember, like how much, if you've got more than one of these things stimulated. So remember, they are backwards here. So I'm not sure understanding the chat. From weakest to strongest, strongest to weakest. Oops, I put weakest to, you're right. Okay, I'll change that here on mine. You can change it on yours either way you want. Okay, sorry about that. Okay, so I would keep all of this in mind for essay 4, because, for example, if you have both a low blood volume, which is going to give you this guy, especially a very low blood volume, both of these stimuli, That could be because you are just bleeding and your osmolarity is totally normal. Or you could have somebody who has high osmolarity and low blood volume, and you could see if, say, someone was severely dehydrated, they had high osmolarity, they had low blood volume, you're going to get multiple stimuli again. Or you could just have someone who has minor thirst and they only have this one. So the amounts of ADH vary as to whether you're getting all three of these or two of them, All right, and then this guy too, just remember that the strongest way, this is right out of the first midterm mostly, but also we didn't really, and 103A, we talked about these, but hyperkalemia can cause you to have aldosterone released, which means it can then lead also to having hypernatremia, because the aldosterone will get rid of the potassium and bring in more sodium. All right, so that is, I think, the end, right? What time is it? We're really kind of out of time. If you're struggling with this stuff, I'm going to change these guys again. Then you're welcome to come to the next lab, or if you feel like you didn't finish the last part of this, then you're, again, more than welcome to come to the next lab and work on things, either together or just, discuss it with me. It tends to not have a lot of folks show up, small groups, so I don't tend to put you into these the breakout groups as much, and then over the weekend, I'll be around if you've got questions. I know it now seems like the exam is a long ways off, but this is not easy material, and I would definitely try those problems that I posted before we go through the answers. Try and come up with the answers and understand why the answers are what they are. So then when we get to them, they'll make, it's as I said, kind of like math. If you don't do the problems, you're never going to really know how to deal with it. Okay, so yes, the urinary Physio-X, this one that we just went through, is due today. The acid-base one is due Tuesday. When we come back next Thursday, we can do some review at the beginning of lecture. And then in lab, what I'd like to do is, and some of the lecture be the beginning of the digestive tract. And then in lab, you'll have time to start drawing. I think there were 3 slides. So while you guys were away working, I started loading models and slides into the study, the lab test 3. So if you've got lots of free time and you want to get ahead, some of them are in there. I didn't finish putting all the slides, but you have the slides that you'll be using next Tuesday uploaded already. So let's see what else now. It's lab 23. I'm not sure I understand that. Can't find the submission. Oh, yeah, I think it says Physio-X 10 on it instead of acid base. I think it says Physio-X 10 or Physio-X 9.
So we've got
like
one that says Physio-X 9 handout, and then we've got one that says Physio-X 10.
And one is due on the 24th because I double checked them.
Right. The 10 is due on the 24th, and then
And that's the acid base one. Mm-hmm.
Okay, so that'll be because
It's probably badly labeled. I think it's actually called something.
yeah, because the handout for today is actually Physio-X 9.
That's the version of Physio-X, and I probably just wasn't thinking.
Okay. So turn in these pages under Physio-X 9 today.
That's right.
Okay.
So the one that's due today is the urinary. I'll rename them. Maybe that'll make it easier and call one urinary Physio-X and the other one acid base Physio-X. So that'll be clearer. And that's due Tuesday, the one that you did today, the acid-base one. We can go over it again on Tuesday in the lab just to make sure you've got the answers. Okay. All right. So if anyone didn't finish and needs more time, you're more than welcome to come to the next lab. So I'm going to let you guys, let's stop sharing, let you go. And any questions over the weekend, I will try to address as soon as I can. And I'll see if I can finish making sure all the digestive slides and models, I think all the models are in there now, but the slides are in there too. All right, have a good weekend. And if you'd like, I'll see you in about 10 minutes in the next lab section.